We're all set. All right. It's my pleasure to introduce Professor Pete Piermarini, coming from Ohio State. He's a full professor and associate chair of the Department of Entomology. He's housed out of Worcester, which is about an hour and a half north of Columbus. So he's a good friend of mine, and we started working on the Mount. Well, I worked on the Malpighian tubules with him. But he is also one of our own. He got his undergraduate from James Madison University in Virginia. He then came down here and got his PhD. What was the department? In zoology. Probably the best organism. He worked on fish and particularly stingrays. So pretty cool stuff that I think he's going to introduce us. Then he did two very nice postdocs, one at Yale University and then at Cornell University with Klaus Beinbach. So it's truly my pleasure to introduce Pete Piermarini. I'm looking forward to what he has to say. Thanks, Pete.
Yeah, can everyone on Zoom hear okay? Awesome, so. Okay. Great, well, thanks so much, Daniel, for that nice introduction. It's a pleasure to be back in Gainesville. It's been the last time I gave a talk on this campus was my PhD exit seminar, which, as Daniel alluded to, is a very different system, working on cartilaginous fishes, in this case, stingrays, which have these venomous barbs, which at the time I thought were extremely dangerous animals. But then, about 10 to 15 years later, I got introduced to the mosquito system. And learned that there are actually more dangerous animals on the planet. And so my research has evolved from vertebrates to insects, but the core questions that interested me as a graduate student working with Dave Evans alongside Keith Cho when I was a student, interested in how animals acclimate to environmental changes. In salinity or salt and water balance, those are still, a big motivation of the basic science questions that I'm interested in pursuing. And that's what led me into studying mosquitoes and their aspects of their renal physiology that I'm going to focus on today. And working on the species yellow fever mosquito, as many of you know, is an important vector of dengue fever, Zika virus, as well as yellow fever, and that working on this species has opened up some new avenues for applied physiology or toxicology related to pesticide discovery, and so that's led to some new fun research directions, allowed me a chance to work with Daniel on some collaborative research, but feeling nostalgic and want to pay homage to my roots as a graduate student. I decided to make this talk more focused on the basic physiology of the mosquito. So I hope you'll learn a little bit about Malpighian tubules, the mosquito kidneys, and share with you some of the different approaches my lab uses for trying to understand some of the basic physiology. And as Daniel mentioned, I'm part of Ohio State University, based on the Worcester campus, and I assume most of you don't know where Worcester is. I didn't know about it until I interviewed there. We're about 90 miles northeast of Columbus. This is the main campus. I know there's a few Buckeye alumni here, so hopefully you'll come back, visit us for the upcoming ESA meeting in November. November is hit or miss weather-wise, but anyway, Columbus, as you know, it's a big city. We have a big campus in Columbus. So one of the most notorious rush hours in the country. And if you come for the conference, maybe you'll be lucky to catch a Buckeye game. But if you mute the mic, I'm still more of a Gators fan. Swamp is still my favorite place to watch the football game. But anyhow, it's a good experience if you coincide with a home game. And I would say for Worcester, if you come to visit Worcester, it's got a different vibe to it. But anyway, it's a really nice place to be. This shows you an aerial view of our campus. So we're in the agricultural hub of the state. So a big, expansive campus, a lot of green around it. So it's a nice place to work. We have representative departments from the College of Ag, including plant path, animal science, as well as entomology, and that building, the Worcester Science Building, is where entomology is housed. And it's a brand new building as of 2021. So coming out of COVID, it was a really nice transition to start working again in a state-of-the-art science building. So anyway, if any of you come to Columbus for the conference and want to make it John up to Worcester, it's about an 1.5 hour drive. So let me know. I'd be happy to give you a tour of campus before or after the conference. So anyway, so that's just kind of my little plugs for our campus. Now I want to get into the kind of the research angle of my talk. And so I know there might be a few non-entomologists in the audience. I want to make sure that and everyone starts off with the same foundation. But mosquitoes, as many of you know, are holometabolous insects. They go through a complete metamorphosis during their life cycle. The larval stages are aquatic, typically freshwater or kind of brackish water at most. They go through 4 instars, where they're just feeding on detritus, organic matter, and pose no direct threat to us. And then after about 5 to 7 days, they usually will go through a pupal stage. They transform into that pupal stage, which is still aquatic, undergoes metamorphosis into the flying terrestrial adults that everyone's more familiar with. And as a reminder, it's only the adult females feed on blood as part of the reproductive cycle. So they need those nutrients in the blood to develop their eggs and reproduce. And because adult females will take multiple blood meals during their lifetime, they have opportunities to acquire pathogens from infected individuals and then transmit them later on, which has kind of led to that moniker as being the world's deadliest animal, given some of the terrible diseases that they transmit. And again, cartilaginous fish are kind of lower on that totem pole. I thought I was working on really dangerous animals as a grad student, but now it puts it into perspective. But anyway, but what attracted me to this system is kind of from an osmotic regulation perspective and ion balance. Going through this amphibious life cycle, the animals face some really opposing challenges to salt and water balance when they go from the larval to the terrestrial stages. And so just to kind of review that, in the larval stage, they're constantly drinking water, typically accidentally as they're ingesting food. There's an osmotic gradient for water to go from the ambient environment into their hemolymph due to the higher concentration of salts in their hemolymph. And at the same time, you have these forces of water coming in. There's the diffusive losses of sodium chloride and other salts to the environment. So that's the kind of the challenges they face as aquatic larvae. And then when they come out as adults, it's kind of the exact opposite in terms of water stresses. Now they're living in a potentially desiccating environment, so where there's evaporative water losses from the hemolymph to the surrounding ambient conditions. So anyways, I found that interesting from an osmotic regulation standpoint. And what's relevant to my talk today, the kidneys of the mosquitoes kind of play some key roles in overcoming these contrasting challenges. So this is going to be the one audience participation part of my talk. So I want to put yourself in the mode of being a larval mosquito facing these stresses. So how would you want your kidneys to work in this situation to deal with these stresses or challenges? Do you volunteer? So this is similar to what's happening maybe after coffee hour. You want a lot of urine or not much urine?
A lot.
A lot, right? So the kidneys of the mosquito, they kind of produce a high volume of kind of dilute urine. So they're trying to they help get rid of that excess water. And at the same time, it's not shown here, but they were trying to hold on to the salt. So reabsorb that salt so that there's, if they could, in an ideal world, excrete distilled water, which is impossible, but that's kind of the idea. Okay, what about in this situation?
A little.
What's that?
A little urine.
Right, exactly. So terrestrial mosquitoes, at least non-blood fed, you're getting little drops of urine maybe every so often. So anyway, so the kidneys have to play roles in controlling how they function to respond to these stresses. Now, what's really cool is if you take a non-blood fed mosquito and let it feed on blood. Within just a matter of a few seconds, now you have an insect that is gorging itself on a fluid that's rich in sodium chloride. I'm not showing it, but also potassium chloride from the blood cells, and a lot of volume. And in just a few minutes, she will increase your body size about 2 to 3-fold in body mass. So I mean, think about that when you go to lunch trying to eat your own body mass in one sitting, right? And this is 2 to 3 times. And then she needs to fly away, right? She has someone she's just stolen blood from who's probably not happy, needs to fly away to survive if they want to lay eggs. So you have a mosquito gorging on blood. So what would your kidneys want to do in that situation?
Get rid of it all the water you can.
That's right. So in a matter of a few seconds, maybe a minute, you go from that, just shooting out a little bit of urine to this dramatic referred to as a postprandial diuresis. And this is, if you scour the web enough, YouTube videos, it'll unfortunately go in your search history, but if you look for diuresis or mosquito pee, there are a lot of images, there's a lot of interesting videos. This is one of my favorites. So here you see an adult female sucking on blood, and this is what we refer to as adding insult to injury. So not only is she stealing your blood, maybe giving you a disease, urinating on you at the same time. So that's what we refer to as the postprandial diuresis. And so this is, again, this process is driven by the kidneys of the mosquito. So what are these mysterious kidneys I'm telling you about? These are what the more proper term would be the Malpighian or the renal tubules. And these are essentially act-like kidneys in all insects except for one group. This is a good qualifying exam question. Which insect group doesn't have Malpighian tubules? Well, you might want to study.
I was about to say it's a mystery.
The aphids are the only group that's lost Malpighian tubules, as far as we know, which is it, but other Hemiptera have them, but for some reason aphids have lost them, even though they do one of the most dramatic excretory events of making honeydew, but somehow they do that without Malpighian tubules. So that's I guess a topic of another seminar someday. But anyway, mosquito kidneys, Malpighian tubules, they are multifunctional. So this is kind of a term that was used when I was in graduate school talking about fish gills. They perform several important physiological roles. So they're involved in osmotic and ionic regulation. That's going to be the angle I focus on today, but also be aware that they're involved in nitrogen excretion, xenobiotic detoxification. So if you're interested in pesticide resistance, Malpighian tubules are likely playing an important role in that. And even in other systems that are involved in Drosophila, they're known to be involved with immune function. So very, very cool tissue. Again, my bias is going to be focusing in on salt and water balance. If you're not familiar with the anatomy of this system, we have here, this is the alimentary canal from adult female. Here's the midgut. This is where that blood meal will accumulate. And then the Malpighian tubules are these 5 structures shown in black in this image. So they have a blind end. So this is a blind ended structure. Urine would kind of flow in this direction to the gut and eventually be expelled by the hindgut and rectum. There we go, sir. And this is a kind of just an illustration showing you what the blind end looks like. And it shows you the cellular composition. There's essentially two main cell types throughout this epithelium. So it's a much simpler system than the mammalian kidney in terms of cellular composition. You have these large, what are referred to as principal cells that kind of wrap around that circumference, forming the lumen. And then you have these smaller stellate cells, which have a nucleated cell body. And then these arm-like extensions, usually have about 3 to 4 that intercalate themselves between the principal cells. And that's really most of what, for mosquitoes at least, that's the cellular architecture. And just to review some basic physiology, the way that Malpighian tubules produce urine is very different than the way our own kidneys produce urine. So on the left here shows you a mammalian nephron. That's a closed circulatory system with a glomerulus. So you have a high pressure that filters the blood to produce urine. The blood cells get left behind, and then the urine goes on its way through the rest of the nephron. In insects, the way that urine is produced is through a process called transepithelial fluid secretion. So this is more similar to the way that our sweat glands and our salivary glands produce fluid. So the way that this works is that here you see a cross section of a Malpighian tubule. Here's a big principal cell, has a brush border with mitochondria, suggesting it's a site of active transport. And then here's one of those arms of the stellate cell, which is much thinner, smaller cell type. And so the kind of the real simplified version of how this works is that you have cations in the hemolymph, sodium and potassium. They are actively transported from hemolymph into the lumen. And then by moving these cations, that creates a favorable electrochemical gradient for chloride to follow passively. And so you get this accumulation of salt in the lumen, and then that creates the osmotic gradient for water to follow. So you end up producing a urine that osmotically is very similar to the hemolymph. And it's referred to as a two-step process. So movement of salts followed by osmotic movement of water. So that is a very simplified version of how this works. In the I guess I say in the next few slides, I'm going to give you a more detailed version, but we're going to be focusing primarily on mechanisms of potassium and sodium. There's, for the sake of time, there's all interesting stories related to chloride. If you're interested in aquaporins, water goes through those, but I'm not going to be going into much detail about them. So just note that caveat. But here is a schematic of how that epithelium works. This is the lumen on the left. That's where the urine would be deposited. This is hemolymph on the right, and we have our two cell types, principal, stellate cell. So in addition to differences in size, they have kind of different biochemical makeups. You have principal cells have a proton ATPase on the luminal side, which is very important for energizing a lot of the transport. On the other hand, the stellate cells have a sodium potassium ATPase on their basolateral side. So just note those basolateral is the side facing the hemolymph, apical is the side facing the lumen. So they do things a little bit differently, but collectively they work together to move ions and water across the epithelium. I know these things can get kind of a little bit overwhelming, but I'll try again to kind of simplify things. So just note that there's multiple ways that ions can be moved across the epithelium, multiple mechanisms. But in a nutshell, you have what are referred to as cation uptake mechanisms on the basolateral surface that would bring cations into the cytosol. And then you have complementary mechanisms on the apical side that will deposit it into the lumen. And so then it worked together to kind of generate that osmotic gradient for fluid secretion. And I would say in my lab, we spent a lot of time trying to kind of decipher, elucidate some of these molecular mechanisms or biochemical mechanisms and how they contribute. And one of the problems, when I started at OSU, we spent a lot of time focused on inward rectifier potassium channels or KIR channels. And we identified two particular genes, KIR1 and what we call KIR2B, as playing major roles. So they account for the majority of potassium that is transported across the epithelium. KIR1 especially appears to play an important role. And kind of just as an offset, kind of some of the work, things that we found, this is some of the collaborative work we did with Daniel and Jared Denton at Vanderbilt, is that if you treat mosquitoes with some of these inhibitors of KIR channels, you can generate some pathologies that at least are consistent with kind of impaired renal function or excretory function. So consistent with disrupting Malpighian tubule function. So this is one of the more dramatic ones that we observe where the abdomen just swells up. And literally you can see sometimes this will literally get to the point of exploding, which is pretty wild to see. And this is in a larval mosquito. Still don't know what exactly is going on here, but it appears to be constipated, which could be a result of not having enough fluid in the alimentary canal through the Malpighian tubules. But anyway, it appears to be some excretory defect with kind of the frass kind of just sticking together. So anyway, that was just some examples of it. You can potentially exploit some of these mechanisms for maybe new mode of action, pesticides. And if you want to learn more about this, put a plug in for a review paper that Daniel and I co-authored with Jared Denton, which kind of goes into the nitty gritty. Also talks about some other roles of Kehr channels in insect salivary gland function and neurological function that Daniel's done some really nice work on. So in his lab, so I'd encourage you to check that out if it's of interest. What I want to talk about today is focus on sodium transport, which compared to potassium movements isn't as well characterized. So some molecular mechanisms of how sodium transported from the hemolymph to the cytosol is really there's not many known mechanisms. There's one study suggesting a sodium hydrogen exchange or NHE is involved, but quantitatively, it's unclear, how much of that sodium moves through that mechanism. But there are some kind of classical physiological studies and pharmacological studies from the 80s that from Klaus Weinbach's lab that found that there's evidence for a sodium conductance. This was pre-molecular era, so they just knew it was inhibited by a blocker called a milleride, which is a generic sodium channel or sodium transporter inhibitor. And there's also some evidence for bumetanide sensitive, what's referred to as a sodium potassium chloride co-transporter. But just activity consistent with those mechanisms, but really what was behind that has never been kind of figured out. And so, and I should also say that there's evidence for cyclic AMP to play a role in activating or stimulating those mechanisms, but otherwise, there's really no insights into what was the molecular mechanism involved. And so that's one of the areas that motivated us to start a collaboration several years ago with Chris Gillen's lab at Kenyon College, which is just about an hour away from my campus in Worcester. And I had a postdoc in my lab, Megha Kelsey. She actually did her master's at UVF in entomology. So she contributed to this work that I'll tell you about today, as well as some of the undergraduates at Kenyon and Chris's lab. So anyway, I just want to acknowledge our funding sources that we've had for this work. And anyway, so when we started this project several years ago, we had our hypothesis going in was that this sodium potassium chloride cotransporter, or NKCC, we just hypothesized that's probably based on the literature, some transporter in that kind of general area or type of transporter is contributing to fluid secretion and sodium chloride uptake. But when we, sorry, I should back up a second. But anyway, an NKCC is potentially involved. But I want to give you a little more background on what an NKCC is and the type of transporters. This is these are called cation chloride cotransporters. So if you're a transport guru, this is SLC12 superfamily. And they are typically called electroneutral transporters of sodium and/or potassium and chloride. And they come in a few different flavors or varieties. There's the sodium independent batch, which are called KCCs. They just move potassium and chloride across membranes, so 1 for 1. So that's why they're electroneutral, one positive, one negative charge. The other variety are sodium coupled, and they come in 2 different types. The NKCC that I told you about earlier, and then ones that are called NaNCC or sodium chloride cotransporters that just move sodium and potassium or chloride without potassium. The other kind of common features of these transporters is that they're typically inhibited by loop diuretics. These are chemicals, the bumetanide, furosemide, and thiazide. And their activity can be modulated by cyclic AMP or protein kinase A. So they kind of fit, that's kind of part of why we hypothesized that NKCC was evolved based on what we knew from the literature and the pharmacology of some of that transport in the tubule. So when we started, we kind of laid out, did a kind of a rudimentary phylogenetic tree of these transporters, looking at mosquitoes and Drosophila compared to the human versions. And just to kind of orient you, here the KCCs kind of fall in their own branch up here that's distinct from the sodium coupled transporters. Here are your kind of conventional NKCCs, human NCC. And what kind of struck us was that there was this other kind of subclade of the sodium coupled ones in insects that we named the CCC2 and CCC3. So we weren't really sure what they were. We assumed that they're involved with sodium transport, but they don't have the same ancestry as the human NCC. So anyway, given that we had some kind of limited resources for our project, we kind of ended up focusing on this group because we thought, there hasn't been really much work on that group, and these kind of conventional NKCCs are probably aren't going to be too surprising in what they do. So we started trying to figure out what these transporters are doing in the Malpighian tubule, and whether or not they could be involved with basolateral sodium uptake. Assumes that they're actually expressed there and they're localized to the proper membrane. And so we did some data mining of an RNA-seq library from the tubules of Aedes aegypti from Emo Hansen's lab. And we noticed that the CCC2 messenger RNAs are, comparable or even maybe sometimes higher in abundance than your conventional CCC1. So both for larvae and adult females. So anyway, that's at least, so they're expressed there. We did some qPCR as well that was kind of consistent with that notion of them being expressed. There's something really interesting going on in the hindgut that is going to have to be we haven't really dug into that yet, but just wanted to point that out as there probably is something cool going on in the hindgut in particular. But anyway, both CCC2 is found in the tubules on the messenger RNA level. And then we developed an antibody specific to CCC2. And this green labeling shows you where that protein is the immunoactivity for the protein. You can see it's occurring on that basal surface of the Malpighian tubules. And this is occurring only in the large principal cells. So you can see the big fat nuclei, those are the principal cell nuclei. And so you can see that labeling on the basal surface. So if we kind of at least pause right now, go back to our model, we at least have confirmed that it's expressed in the tubule and it's on the proper membrane to have a potential role in sodium uptake. But what are the functional properties? It's kind of in its own little part of the phylogenetic tree, so doesn't quite fall into either category cleanly. So we hypothesized that, at least given that it falls within that broader sodium dependent clade or sodium coupled, it would have transport properties resembling a conventional or maybe one of these NCCs. So how do we go about determining that? Well, we use a system. It's called heterologous expression. So we express our proteins of interest in frog oocytes. So if you're not familiar, frog oocyte is Xenopus laevis. They're large cells, about 1 millimeter in diameter, so it makes them kind of relatively easy to work with in terms of doing electrophysiology or other physiological bioassays. And what's nice about these cells is that you can inject them directly with the messenger RNA of interest, which we refer to as cRNA in this context. And then within a few days of injection, they start translating that messenger RNA into protein. And oocytes tend to be very good at taking a foreign messenger RNA and properly expressing it to the cell membrane for an ion transport or a channel. So they're a nice system to work on. So we can get our protein of interest on the plasma membrane, and then that allows us to assess what its functional properties are. And going into this, again, we assume that we're dealing with an electroneutral transporter. So the first way of measuring function is something called ion uptake assays. This is where you take referred to as a tracer ion. So we use rubidium, which is known to be able to substitute for potassium in many transporters, and then lithium, which is known to substitute for sodium. And so we can measure the accumulation of these cations inside the cell to get an idea of rubidium or of potassium and sodium transport. So what did we find? So when we take the CCC2 messenger RNA, express it in the oocytes, well, what we found was that lithium uptake was much higher than our control oocytes. So for controls, we just inject them with water, like distilled water is a control. And so you can see that lithium uptake is substantially higher, almost tenfold higher in our CCC2 oocytes. So that makes sense in terms of potential sodium transport. But when you look at rubidium, interestingly, it's no different than the controls. So if you kind of try to interpret this first finding, we kind of thought, well, if it's moving lithium, not rubidium, this would be consistent with sodium chloride cotransport, right? It doesn't need potassium. So the way that we would test this hypothesis is to remove chloride from this extracellular fluid or bath. If this is an NCC, it would need chloride to transport the sodiums. And so that was the next experiment that we did. And again, we have lithium uptake on the y-axis. Here are the control cells, and then here's our CCC2. And you can see that removing chloride had absolutely no impact on the lithium uptake. So very mysterious. And then we threw some of your typical loop diuretics at the oocytes, saying, well, let's see if we can at least block it with something that's known to block CCCs. And you can see this is our vehicle, and there was no significant inhibition. And these are concentrations that should annihilate transport activity if it was just a conventional NKCC. So we kind of step back again. It appears to be, this transporter appears to be mediating sodium movements without chloride. Then anyone know what could do something like that? If you're moving just sodium by itself, what would you usually do that? Sorry. Yeah, it's kind of like a pump type that where you're just moving one ion. So, or in this case, like a channel. So it's just allowing the movement of, because ion channels, can be selective for sodium or potassium. So if you're moving sodium without any anion, like chloride, that would be something like a sodium channel. So anyway, which is kind of heretical for this transport. No one's ever, I don't know if anyone's tried to test that before in this family, but anyway, that was our best guess, was that it must be operating like a sodium channel. So how do we go about demonstrating that or testing that? So we use a technique, again, going back to the oocyte, it's called 2-electrode voltage clamping. So you impale the cell with 2 electrodes that allow you to measure or control the membrane voltage, and then if you clamp the cells at a voltage, that's done by injecting current. So if you have a protein or channel that allows the passage of electrical current, like a channel would, you can detect that activity using this type of approach. So, and this just shows you, this is kind of a lot to take in, but it's called a current voltage plot. So one of the first things we did was we clamped, the x-axis shows you where we clamp the voltage of the oocytes. So you do a rapid change of voltage. And then what you look for is a corresponding change in current. If you have cells that do not have much electrical activity, if you connect the dots, it looks more or less like a flat line, a low slope. But what we noticed with the CCC2 oocytes is that they kind of tended to kind of bend downward to have a steeper slope, especially at more negative voltages. So that's some activity consistent with a sodium channel, because as you make the cell inside more negative, you're bringing more current into the cell. So positive charges are moving in. But this was done in a physiological saline, so it had high sodium, low potassium, so it's kind of harder to discriminate what's going on. So we did another set of experiments where we just clamped the oocyte at a constant voltage, a negative voltage, below its normal resting voltage. And then we, one at a time, added different cations to the bath. And so what you want to look for are these downward deflections of current. And you can see that when we add potassium to the bath, we see very little change, but lithium and sodium give these really dramatic downward movements of current. So that is consistent with a channel that is selective for sodium and lithium over potassium. So that's kind of how we can potentially explain those ion uptake results. And this is again a control oocyte that shows very little changes, if any, to the similar treatment. So from this we essentially concluded that instead of being one of these canonical SLC12 transporters, the CCC2 insect unique subclade is acting more like a sodium channel, which is very, very weird for this. Again, no one's described this activity in this family before. And I don't know how many people have actually tried to look for it, because they've always been assumed to do electroneutral co-transport. So if we go back to our transport model, if we put together the localization, functional characterization data we have here, identified a potential mechanism on the basolateral membrane of the principal cells that could mediate the uptake of sodium from the hemolymph. Now, as I mentioned earlier, there was earlier pharmacological studies suggesting that conductance could be activated by cyclic AMP, maybe inhibited by amiloride as well. And in terms of the amiloride pharmacology of the channel, this say that at the moment, we don't really have any idea how to block this channel. Amiloride doesn't appear to be doing it. But in terms of cyclic AMP activation, we have had some luck going down that route. I wanted to first show you kind of what the original data was in the Malpighian tubule. So this was from Klaus Feinbach's lab in the 80s, where they impaled individual principal cells of Malpighian tubules, measured the membrane voltage, which is on the y-axis. And what they found was that when you add cyclic AMP to the bath, the membrane voltage becomes more positive, which is consistent with activation of the sodium channel. And that was the interpretation of that evidence. So one of the first things we tried with the oocytes expressing CCC2 is seeing how does their membrane voltage respond to cyclic AMP treatment. And what you see here is a representative tracing, and you can see that the trajectory of membrane voltage is very similar to what was described in the Malpighian tubule principal cell. Although the kind of the magnitude are going to be different because you're working on different cell systems. So anyway, this is something consistent with an activation of a sodium channel. But to kind of confirm that, we did more of the voltage clamping experiments. And just to, again, we're looking at how the currents change across different voltages. And sorry, I should have pointed out this is, what you're going to be seeing is the current voltage plots at before cyclic AMP and then 30 minutes after the treatment. And so you can see here the black data points are the before cyclic AMP, the red is after. And so you can see again a further downward shift in the currents that are being mediated through that channel, consistent with cyclic AMP activation. And then this is again this water injected cell where you can see there's kind of nominal effects of that same treatment. So, so anyway, so our kind of conclusion as to where we are right now is that the CCC2 appears to be consistent with a cyclic AMP activated basolateral sodium conductance in principal cells. There's still a lot more work to be done before we can probably carve out what its exact role is in fluid secretion. Because it would be really nice if we had a blocker of this transporter so we could do those classical Ramsey assays that you might have learned about in insect physiology class to measure their effects on the tubule function. And also kind of more questions related to, what role is it playing? Is it playing a role in the diuresis after blood feeding? So there's still a lot of questions, but I think we've at least uncovered a new potential mechanism that may have some importance, especially when you consider that, as I showed you in that phylogenetic tree, it appears to be something novel to insects. So potentially for pesticide development, you could develop maybe insect-specific blockers of that channel, assuming that it might kill them if you inhibit the channel. But anyway, just to kind of summarize some of the take-home points, hopefully kind of learned today something a little bit new about Malpighian tubules and how they play key roles in countering those challenges to water balance that mosquitoes face as part of their amphibious life cycle. And that the Malpighian tubules produce urine through kind of the coordinated activities of those two different cell types that is mediated by kind of numerous transport mechanisms. And I just kind of focused in on one, the CCC2, which kind of excitingly appears to be a novel transporter in terms of its function that has these unconventional channel-like properties. And from what we've uncovered, it appears that it may play some role in that cyclic AMP stimulated sodium uptake in principal cells. So with that, I just want to thank you for your attention, and if you have any questions, I'd be happy to answer them. So thank you. Hey, Dave.
Yeah, very cool talk, Pete. So in the human kidney, we have the epithelial sodium channel on the apical membrane, the ENaC channel.
Right.
Do you think that that's just potentially flipped here and that we're annotating this as a CCC, but we might be looking at more like an ENaC-like channel that is insensitive to a millerite, but essentially.
Okay. Yeah, I don't. Part epithelial sodium channels in insects. I don't think there's a direct ortholog of like that renal ENaC. There's the pickpocket genes, which I think play roles in the nervous system. As far as I know, the pickpocket expression in the tubules is fairly limited. But I think it's probably analogous functionally to how those channels would work, because that proton ATPase on the apical side creates a really strong negative potential inside the cell. So any basolateral channel on that allows cation movements the cations are going to be pulled through those channels by that VATPase. So in the human kidney, it's kind of you have the apical sodium channel with the basolateral sodium pump that kind of moves that along. But the CCC2 is, if it evolved, I don't know. We have to look at maybe structurally, does it have any similarities to ENaC? That would stand out. But as far as we can tell, it's just weird. The one things we have noticed is that in the mammalian literature, it's pretty well known which domains of the protein are involved with like potassium and chloride movements, as well as bumetanide sensitivity. And those parts of CCC2 are all kind of disrupted compared to the mammalian ones. So at least on the amino acid level, it's consistent with being something different than your traditional NKCC. But yeah, thank you. Yeah, Keith.
Maybe, yeah, no, nice talk, because cell models bring back a lot of memories from PhD times. So, yeah, as you say, like that's really interesting, divergent finding for this family, right? And so,
Right.
and my ability to read the electrophysiology is limited. So can you tell from your experiments, can you distinguish channel versus facilitated diffusion? And then if you, in the sequence alignments, are there some clues about what's different that's allowing it to act so uniquely relative to the rest of the family? Yeah.
Yeah, so that's a good question. So so yeah, you can't, in the voltage clamping setups, you're not gonna pick up facilitated diffusions. So those are, so that's why the VM, just changes in membrane potential, those could be due to, like if you raise potassium on the outside of the cell, it's gonna depolarize the cell. And that's not due to channel-like activity, that's changing the electrochemical gradient. Yeah, by voltage clamping, you're kind of, the only change, the changes you see have to be done by electrical, either a channel or like an electrogenic transporter. And so that's, I mean, and that's, there's like, kind of looking at the difference between a channel and electrogenic transporter, there's, in the literature, there's kind of arguments over, what makes a channel versus a electrogenic transporter, electrogenic, and I don't think there's like a clean, there's people that'll argue that, electrogenic transporters actually are channels. But yeah, but as I was mentioning, in the terms of the protein structure, the amino acid sequences, yeah, the areas that are, at least in the mammalian system, mammalian transporters associated with potassium binding, chloride binding, and bumetanide sensitivity, they're not well conserved in this one, but the sodium binding area is well conserved. So, and some unpublished stuff that we're still working on, we've done some mutagenesis in that sodium binding area. We appear to be able to knock down the channel activity, which was kind of key because, because you always have a kind of a doubt in the back of your mind, sometimes, especially when you see something so weird compared to what everyone in the literature's found. But that was kind of a relief that we could actually block the channel activity by mutating it. I should also say, I didn't share, is that in Drosophila, we've also characterized the Drosophila ortholog, and it has this essentially the same functional properties. So it's not just unique to the mosquito CCC2, it seems like, at least in flies, at least, this is a common, this channel-like function. But I mean, we still can't rule out that it's maybe electrogenic there's some other anion being moved that we just haven't tested yet, but that's becomes probably a fishing expedition. You just have to screen stuff. Yeah, it's a good question. Yeah.
Just to piggyback right up what you were saying.
Yeah.
The mutagenesis where you were to knock down, did you see any perhaps mortality when you did these kind of mutagenesis screens? If you did knock it down, there was altered survival?
No, sorry, we did the mutagenesis with the protein.
Oh.
So in the functional
within the
right. Yeah, we didn't mutate the mosquito or the flies. Right.
You don't know this. Has there been any attempts made to get the structure of this case, this CCC2?
Yeah, we've done. Yeah, Chris's lab, they've done some of the alpha fold to kind of try to build a rudimentary model. Yeah, and that's what kind of allowed us to kind of identify which amino acid residues might be important in the sodium channel activity. But yeah, we're still kind of working that. That was a lot of work that his lab did, so I left it out from the talk. But yeah, but it does appear to be you can, if you mess around with that sodium binding area, you can mess up the channel activity, which is encouraging.
Yeah, thank you very much for your talk. I learned a lot for my video and very event. I have two questions. The first one is what's the source of cyclic AMP? As far as I know, the Bartigan tuber can be activated by so many different kinds of neuropeptides, and upstream of neuropeptide, they may have the increase of cyclic AMP, like a kinine or calcitonin and so on.
Right.
So that is my first question.
Okay.
And second question is, have you done for the RNAi for the CCC2 and if yes, what's the phenotype? And if no, then what do you expect with the phenotype?
Gotcha. Yeah, so the first question, the cyclic AMP, yes, I didn't really get into the signaling there, but cyclic AMP, there's a hormone neuropeptide called calcitonin-like peptide that there's receptors for in the principal cells. And when that peptide binds to the receptor, it triggers the cyclic AMP pathway, PKA pathway. So and that contributes to that postprandial diuresis that I talked about earlier. And so, yeah, so the physiology of the tubule, when you activate it with cyclic AMP or one of those diuretic peptides, it activates a whole suite of transporters. So the proton ATPase gets revved up, and that kind of helps bring more things across the epithelium through some of these transport mechanisms to help produce more urine. And then the second, RNAi is stuff, I don't know, I have trouble with RNAi. Whenever I try to knock something down, the phenotypes are pretty boring. And so we haven't really done after it. I probably talked to Chris. I know Chris's lab's done more of the RNAi, but sometimes with mosquitoes, but you have this other ortholog, CCC3. So whenever we don't see an obvious phenotype, you don't know. Sometimes you get compensatory expression of other things. CCC3 is not heavily expressed in it. It's more specific to larval mosquitoes. But we don't know. My lab hasn't tried doing injections with the CCC2 double-stranded RNA. So yeah, unfortunately, I don't have any insights to that, but I don't know. It's hard. Like I showed you earlier, the hindgut is also a really hot spot of expression. So I don't know if we would have to knock it down and then do probably Ramsey's so we could isolate the tubules and see what the effects are. But yeah, I don't know. That would be really interesting to work on. But that's why the pharmacological, if we could find a pharmacological blocker, just nice, because then we could pull a fresh tubule out, hit it with an inhibitor, and then don't have to worry about compensatory effects like in terms from RNAi. But
Thank you.
Yeah. Sorry.
I also have two questions. That's a great talk. So, first one is, have you looked at the CC2 expression file difference before and after blood meal? And the second question is, do you think the molecular CCC2 is primarily driving the sodium entry for secretion, or it's kind of like a modulatory role that can stabilize the osmotic pressure during dialysis? Second, does it make sense?
Yeah, so the first question, what happens after a blood meal? I don't know. I know in general, after a blood meal, you see genes associated with diuresis are downregulated because the diuresis is usually mosquitoes come in kind of primed, adult female are primed for diuresis. So genes that are important for diuresis tend to be upregulated in non-blood fed mosquitoes so that when they feed, they can pee it all out. And then you see a downregulation over time as they start kind of transitioning from a diuresis state to more of a detoxification state later on. So yeah, we've done some RNA-seq showing that. Most of those diuresis genes are upregulated. So I can't remember with CCC2 what happens, but if it does anything, I would expect it probably to decrease after blood feeding. But I don't know for sure. And then what was the other question was, does the channel?
Yeah, is it primary sodium like the transporter for secretion, or it's just the modulatory role with the osmotic pressure stability during the diuresis?
Okay, yeah, I don't know. That's a thing. Yeah, it's hard to isolate because sometimes there's a lot of overlap, in cell volume regulation, and so yeah, so it's hard to know for sure. And there could be kind of redundant, could be involved in both, or one or the other. But yeah, I don't have an answer to that, but yeah, but it's a good question. That sometimes these, especially when you're looking at transporters that are involved with both salt and like, proton movements. It's hard, like, is that transporter functioning for sodium or is it transporting for pH regulation? You know, it's doing both, it's probably both, but it's hard to isolate. It kind of depends probably from your perspective. And if you're interested in acid-base regulation, you probably think it's more involved with, you know, with that. If you're in cell volume regulation, you might think it's involved with that, but reality, it's probably doing a little of both. But yeah, so I think that's a good question.
I think we have chance for one, just
Yeah.
that's one, quick one.
I got one.
Okay.
Yeah. Thank you for the presentation and for coming down from the frozen north that is Ohio. My question is, I didn't see any like brackish or saltwater mosquito organisms in that clay that you had. Do you have any hypotheses as to the expression or potential expression for some of these sodium pumps or sodium channels for that?
For like brackish water species.
Is that
Yeah.
also a direction you want to move into, I guess?
Yeah, I think it's a really good question. There's not. The only work that I'm familiar with on adaptation to higher salinities is with anopheles. There are some subspecies that have
Albumin.
salt tolerant. Yeah, they're salt tolerant. And some work out of Notre Dame where they've kind of done SNP study looking at kind of selection. And I know there's like sodium hydrogen exchangers where you see kind of evolutionary changes in those genes. I'm not sure. I should check if they found it in some of these. Cause yeah, it would be interesting if they're playing a role. Yeah.
Rhinincus.
Right. Yeah. And some of those salt tolerant ones, that would be really cool to look at. Yeah.